Hello, and thanks for joining us for today's webinar, Measuring Innate and Adaptive Immune Responses in Ruminants. I'm Amanda Turner, a Product Manager at ABD Serotech, and I'm here to moderate the event. I'm pleased to introduce Professor Gary Entrican as your presenter. Professor Entrican is a Principal Research Scientist at the Moredun Institute and holds an Honorary Professorship within the College of Medicine and Veterinary Medicine at the University of Edinburgh. He also holds the chair of the IUIS Veterinary Immunology Committee and is a member of the steering committee of the BBSRC UK Veterinary Vaccinology Research Network. Gary will present novel, recently completed work carried out by groups at the Moredun and Roslin Institutes in Edinburgh, giving us an insight into the very important area of defining and measuring correlates of protection. Following the webinar, we'll have a Q&A session, and we welcome any questions you'd like to ask. At any time during the webinar, you can click on the Q&A button at the bottom and submit your question. We'll answer these at the end of the presentation. If a fuller answer is needed or we run out of time, your question will be answered later by email. An on-demand version of the webinar will be available to watch again via the ABD Serotech website. If you experience any technical difficulties, please visit our webinar help guide by clicking the Help button below the presentation window. It has a question mark icon and covers common technical issues. I'll now hand over to Gary.
Thank you, Amanda, for that very kind introduction and for the opportunity to do this webinar today. What I'm going to do is give you some background and introduction to why we took the approach we did to do these experiments, why we need to have these immunological reagents for livestock species, and how we approach this on a global basis. I'm then going to go into some of the science, which is how we defined ruminant myeloid subsets using CD14 and CD16 as markers of subpopulations, and how we then define those populations in cattle and sheep, and then look at some aspects of adaptive immunity, and in particular how we define the Th17 subsets in cattle and sheep. And then I'll finish up with a summary and acknowledgements. So the project that I'm going to describe is a BBSRC Industrial Partnership Award with Bio-Rad ABD Serotech. This project started in 2012 and recently completed at the beginning of 2015. And you can see from the title, what we were aiming to do here was to improve our capability to detect immunological correlates of protection in ruminants for research that would underpin vaccine design. And what we essentially did was, at the time, we did a scoping exercise to identify what the gaps were in our capability. So the objectives that we specified were that we would evaluate monoclonal antibodies for reactivity against myeloid subset marker for myeloid subsets in sheep and cattle. We would then clone and express bovine and ovine T-cell subset transcription factors and evaluate monoclonal antibodies for cross-reactivity. That particular objective I'm not going to describe in detail today, but I will mention how we progressed it towards the end. We also set out to clone and express bovine and ovine IL-17A and evaluate monoclonal antibodies for cross-reactivity. And then to compare and contrast the immunological similarities between cattle and sheep. And that was an important component because we felt that we do know that there are aspects of cattle and sheep that often are similar, but there are also some differences. So in terms of how we apply immunological correlates, what we're trying to do is develop a good and solid knowledge base for our strategies for controlling infectious diseases of livestock species. And those strategies can be vaccines, they can be diagnostic tests, and in particular there's a strong interest in developing DIVA tests, which would be the discrimination between infected and vaccinated animals, and then using these in combination to develop new management strategies and also policies for controlling infective diseases that impact on livestock welfare, but also on livestock productivity and agricultural productivity. In terms of the science, what we're really trying to do is reduce the guesswork in vaccine design. So the more that we know about the immune response, then we can target our approaches. That's obviously very important in terms of our usage of animals, because we can then reduce the number of animals that we use in experiments because we've got more targeted approaches. The other thing that this allows us to do is to widen the option of our selection of biomedical models for human diseases. So there are some situations where utilizing small laboratory rodents may not be ideal. That might be for longitudinal studies or for using animals that are larger, and in which case that might be the case that we can use our livestock species. The various elements that we have here are looking at correlates of protection, which would be for vaccines, correlates of infection for diagnostic tests, and correlates of disease, which would help us understand pathogenesis. And what you can see from this Venn diagram is that there are likely to be immune correlates that are going to overlap with them. So there's may not be, and it's unlikely that there will be single immune correlates that are going to fit into each of these boxes, but in the selection of the immune correlate, we need to know where that immune correlate sits within that Venn diagram, and that's why we need multiple immunological reagents to then identify that and tie up our pathogenesis, vaccines and diagnostics. So with this project, we are trying to identify and address the needs of the veterinary immunological research community. And as I've mentioned, what we want are immunological reagents that will conduct high quality research that has impact. And what we need are really functionally validated reagents that can be used in specific techniques. And I will describe later some antibodies that we've evaluated for more than one technique for identifying T-cell activity and function. It's important that these reagents are both affordable and accessible to the research community, and those elements really then tie into how do you make those, and how do you get the information out to the research community, and also how do we maximize our impact as a global community. And of course, information resources are very important to us. We rely a lot on peer-reviewed publications. I think we are not so good at data sheets and databases, and that's something that we need to develop in the future for our veterinary vaccinology reagents. And we also need a lot more web-based resources that people can access from around the world. How do we achieve this? Well, we really need to be able to collaborate and network so that we can identify our priorities. And I've just used as an example here the International Union of Immunological Societies, where there is the Veterinary Immunology Committee have a subcommittee called the Toolkit. And that has been running now for about 15 years or so. And the ethos behind that toolkit is that it gets people together. We have workshops at the International Veterinary Immunological Symposia every 3 years. What we're aiming to do is avoid duplication of effort, to identify funding opportunities, and also, very importantly, how researchers can engage with industry for the quality assurance, the marketing and distribution of these reagents, which goes back to the earlier point about how we make these affordable and accessible to the research community. I mentioned the IVIS, the International Veterinary Immunological Symposium, and at the most recent meeting in 2013 in Milan, there was a presentation by STAR-IDAZ, and I'd like to thank Sadhana Sharma and Alex Morrow for providing me with the information here on this slide, which they presented in Milan. And they were describing STAR-IDAZ, which is the Strategic Alliance for Research into Infectious Diseases of Animals and Zoonoses, and they conducted a survey of on a global basis where they got 100 respondents from 19 different countries, and they asked a number of questions about what were the barriers to improving our research capability for control of the veterinary diseases and zoonoses. And you can see from this pie chart here that immunology figured very highly, tools and understanding host pathogens and their interactions. And of course, all of those elements depend on our capability to analyse immunological responses. You can see the web link there to STAR-IDAZ, and I'd recommend that you have a look at that, and you can see what that network is aiming to achieve. It's funded currently through a Framework 7 initiative by the European Commission. Following on from STAR-IDAZ, that information and the gaps that were identified were very important for underpinning the development of the UK Veterinary Vaccinology Network, which is supported by the BBSRC. And within this network, what we're aiming to do is to support the multidisciplinary research community, which looks at various aspects of veterinary vaccinology. And within that, some of the aims are to develop these new technologies and immunological tools which will enhance our understanding of protective immune responses, and, as I also mentioned, help us to understand host pathogen interactions at multiple levels so that we can then develop more effective vaccines. And part of the initiative here is also to support these open source species specific immunological toolboxes, so that goes back to the information exchange that we have with the global research community. And again, I've provided a web link there where you can see the Veterinary Vaccinology Network. The launch meeting for that was in Birmingham in the UK in February this year, 2015. So this is a very early initiative. It's running for 5 years, and we're looking to see how that develops. This just gives you an indication visually of how the Veterinary Vaccinology Network will work. And what you can see is the network really aims to be at the hub of a number of activities which involve collaboration, networking, sharing of information and knowledge, and how we would then apply these to improve our capability to develop vaccines and also develop new technologies, and how we then manage our infectious diseases through novel approaches. One of the important things that we considered when we were doing our research is that we often make assumptions that the immunological paradigms that we know from the literature will always hold true and be consistent across different species. And there's often a very solid base for making that assumption, but sometimes it doesn't follow through. And the way that we know it doesn't follow through is when we have appropriate tools or reagents, it allows us to investigate that. So there may be, for example, cell surface molecules that appear on a cell population in one species that might not appear in the cell population in another. And that, of course, is then very important if you're trying to define the function of that subset, and you can only do that with good and reliable immunological reagents. So if we think about how we're going to analyse these immune responses, and in our case this was in cattle and sheep, we have to think about how responses are induced naturally by infectious agents. And a common route for infectious agents, obviously not in every case, but in many cases, is through mucosal surfaces. And epithelial cells will line our mucosal surfaces. They will be often the first point of contact for infectious agents. When those infectious agents come in contact with the mucosal surfaces, those epithelial cells will respond. And they will respond by essentially sending signals that will recruit other innate immune cells. In this case, I've indicated dendritic cells, macrophages, and neutrophils. And the principle here is that those cells will act, first of all, as a primary line of defence, but also be important for activating the next stage of the immune response. And what we can see here is how the innate immune system begins to interact and bridge with the adaptive immune system through antigen presenting cells, which will interact with CD4 T cells, but also interact with other cells such as natural killer cells and gamma delta T cells that will be involved in that very early stage of the immune response. This then develops further into the subsets of CD4 T cells, and I've just given an example here of some of the subsets. Th1 type cells, which are important for defense against intracellular bacteria, Th2, which are important for defenses against parasitic infections, mainly nematodes, Th17, which are involved in responses to extracellular bacteria and fungi, and of course regulatory T cells, which are very important for controlling these immune responses. In addition, we have CD8 T cells, which are important defense mechanisms against viruses, and of course B cells, which will secrete antibody to neutralize and combat extracellular pathogens. And the main point here is that these subsets don't act in isolation, they interact together, particularly as I mentioned with the regulatory T cells that are important for stopping immune responses and controlling them, but also the feedback back to the innate immune system, for example with the Th1 cells producing cytokines such as interferon gamma, which will then activate macrophages which may have ingested intracellular or been invaded by intracellular bacteria and program those cells to kill those bacteria. So this is a very dynamic system and quite a complex system, and therefore it's important for us to be able to understand this if we're going to develop safe and effective vaccines. For a long time, the innate immune system was, I wouldn't say ignored by immunologists, but certainly wasn't regarded as important as the adaptive immune system. And when we go back to Eli Metchnikoff, who really was the first cellular immunologist, and it was Eli Metchnikoff that noted that phagocytic cells existed and that they would ingest microorganisms and kill them. And people thought that that's just what macrophages did, and that was all they did for a very long time. And of course it was with our ability to analyse immune responses that we began to realise that those cells were much more complex. And what we can see now is, of course, that there are multiple types of macrophages in different organs of the body. These perform very different functions and they have different features. And because of that complexity, if we are going to then manipulate these cells or target them for vaccine delivery, we really need to understand how they work. So at the time that we started this project, we were interested, as I said, in understanding how we could further dissect the immune responses of cattle and sheep. And for the myeloid work, the approach we took was fairly simple and straightforward, which was we would look at CD14 and CD16 as potential subset markers for monocytes in the peripheral blood of cattle and sheep. So CD14 is co-receptor for LPS, and we know from other species, and this is the point I was making about the assumptions we can make, it is expressed highly on monocytes, not so much on granulocytes, and is also expressed by tissue macrophages, but tends not to be on dendritic-like cell populations. CD16 is the low affinity Fc receptor, and this is found on the surface of natural killer cells, neutrophils, monocytes and macrophages, so it has a wider population distribution. again, based on what we know from other species and how these cells are important for immune function, looking at monocytes in the blood, we have the classical population, which tend to be CD14 high expressors, CD16 negative, and express the chemokine receptor CCR2. We have the intermediate population, which is CD14 positive, but CD16 positive low. And then we have these non-classical monocytes, which are low expressors of CD14, high expressors of CD16, and these express the chemokine receptor CX3CR1. And it is thought that these subsets develop into other cells in tissues, and here we have different types of macrophages with either a pro-inflammatory profile, anti-inflammatory, or that they are involved in antigen presentation. And it's important to say here that there are various degrees of how these cells differentiate, and we're still not sure about these lineages, where they originate and exactly how they relate to each other. So we use these terminologies to try and understand them, but this is very likely to be a continuum of populations of cells. So for the analysis that we did looking at CD14 and CD16, the percentages that I'm showing you under the images of the cattle and the sheep represent the percentages of CD14 positive cells that we would normally typically see in the blood of these animals. And the first thing to note is that we see a much higher proportion of CD14 positive cells in cattle than in sheep. And that's always been of interest to us as to why that might be the case. We don't know exactly why it's the case, but certainly if you then start to think about how this might impact on in vitro assays, where you're taking peripheral blood cells and using the monocytes as antigen presenting cells, then that can affect the outcome of those assays. So it's a very interesting phenomenon that cattle and sheep do differ in the proportions of CD14 positive cells in peripheral blood. When we look at the CD16 staining, what you can see is that there are distinct populations based on side scatter and CD16 expression, and the population that does look different between cattle and sheep is that there is certainly a higher proportion of CD16 high positive cells in cattle. When we look at these cells further, based on CD14 and CD16 expression, then what you can see is that the classical population is quite large. That's the CD14 positive and CD16 negative. We then have the intermediate cells, which are CD14 positive, CD16 low. We then have the non-classical, which are CD14 negative, but CD16 high. And then there is a population which are CD14 negative, CD16 low. And I'm not showing the data here, but we know that those cells are NK cells because they express NKp46. When we compare that profile between cattle and sheep, so you can see the sheep again in the bottom scatter plot, then those populations are smaller, and certainly that high population is very, the CD16 high non-classical is very, very difficult to detect. So again, some clear differences between the populations in cattle and sheep peripheral blood. We were interested to know how these might relate to those putative populations in human peripheral blood. We don't have the antibodies to do the analysis for the chemokine receptors or for CD163, which is a scavenger receptor. But what we did was we sorted the cells in cattle. We couldn't do these experiments in sheep because we didn't have enough cells, but we sorted the cells and then did RT-PCR looking for those chemokine receptors and for CD163. And what you can see is that the classical CD14 positive, CD16 negative cells are CCR2 positive and CD163 positive, whereas the non-classical CD16 high are CX3CR1 positive. So that seems to fit the same kind of paradigm that you would expect from our knowledge of human peripheral blood monocytes. What we are of course interested in knowing is what is the function of all of these subsets, and in particular of this CD14 negative, CD16 high population. We know that in cattle these are around up to 1% of the peripheral blood mononuclear cells. Their monocyte morphology, and you can see that in the image, they're CD172 alpha positive, which is SERP alpha, and CD115 positive, which is CSF1 receptor, and CD14 negative. We know that they are not NK cells because they are NKp46 negative. They express interleukin-12, not IL-10, and will respond to TLR ligands by upregulation of IL-1 beta. And they can, on sorted populations, induce greater allogeneic responses. So we think these may be a typical inflammatory type cell, but we still don't know what the relationship is between tissue macrophages and dendritic cells, how they interact with pathogens, and what the role might be in interacting with T-cell subsets. So that leads me to discuss how do we measure our T-cell responses. And traditionally we do use cell proliferation as a marker of antigen-specific cellular recall responses. But that will only give us a certain amount of information. It certainly doesn't tell us about the bias of that CD4 T-cell response, and therefore we need to be thinking more about other methods of looking at our T-cell responses. What we do know is that when we've looked at cytokine responses and compared them to proliferation, then we don't always get the same result. And here what I'm just showing is some published data looking at interferon gamma responses correlated with cell proliferation in activated T cells. And what you can see is here that there is no correlation. So that means that we need to be very, very careful when we are selecting our immune correlate to investigate immune responses. Clearly, the greater the capability that we have, then the more we will be able to dissect that response. So it's not only the correlate that we select, but it's also how do we do those experiments. And again, this is published data, but very interesting in telling us that the time that we choose to harvest our sample and analyze it will affect how we interpret those data. So for example, what you can see looking at interferon gamma responses is that we see a very strong peak of message very early after cell activation, and then this would be followed by protein production, which would gradually accumulate. This is measured by ELISA, so this will gradually accumulate in the culture supinate. In contrast, when we look at interleukin-10, we see that the peak of that response is later, and again the protein will continue to be produced and secreted and accumulate in the culture supinate. And the point there is that wherever you harvest, particularly if you're interested in knowing whether you've got a pro-inflammatory or a regulatory type response, the ratio of those cytokines to each other is going to change over time. So selecting that time point is absolutely crucial. So when we look at the various features of CD4 positive T cell subsets that might help us identify them, how they are activated and ascribe function. What we can see here is that there are a number of transcription factors and signaling molecules associated with different subsets. And of course, the initial paradigm was for Th1 and Th2, and that was the seminal work that was done by Mossman and Coffin back in 1986. And that demonstrated very clearly that there were different CD4 T cell subsets that produced different cytokines. And even though that was done in mice all those years ago, we still don't know exactly whether or not those subsets exist in ruminants and how they might interact with each other. The Th17 subset was described much later, 2005, and because of that, certainly our ability to look at that subset in veterinary species has lagged very much behind, because we've not had the reagents, and people certainly weren't looking for them until fairly recently. So at the start of the project that I described earlier, this was the scoping exercise that we did, and we were looking to see where we had cloned molecules. We had taken an approach at Moredun for quite a long time now of expressing recombinant cytokines in Chinese hamster ovary cells, and this was by way of immunological tools to allow us to ascribe function to those cytokines, but also to act as positive controls for measuring cytokines in other cell populations. What they do allow us to do is definitively ascribe activity of monoclonal antibodies. So do those monoclonal antibodies recognize the product, the gene product that we are interested in? And what you can see is that we had cloned Th1 and the Th2 cytokines, signature cytokines, interferon gamma and interleukin 4. We also had interleukin 10. We had expressed some, but not all of those, in CHO cells, and there were existing monoclonal antibodies. We really didn't have very much at all in terms of interleukin 17, and our knowledge of the transcription factors, our ability to look at those transcription factors, was certainly very limited, and really only to FOXP3, which is the transcription factor for regulatory T cells. So we embarked on a process of cloning and expressing both sheep and cattle interleukin 17A. And what you can see here is that we did successfully express those. You will have noticed from the previous chart that there were some polyclonal antibodies available to bovine interleukin 17A. We did not know if they worked on sheep, so that was one of the first things that we did. And you can see here that those antibodies will pick up ovine, recombinant ovine interleukin 17A by ELISA. Interestingly, they also pick up native interleukin 17A produced by mitogen-activated sheep T cells. Those antibodies can also be used in an ELISpot, and again, this was one of the points that I was making at the start about evaluating reagents for multiple assays, so that we can not only look, in this case, at the secretion of interleukin 17A in cultures, but we can identify the frequency of interleukin 17A producing cells. So you can just see here, again, these are CON-A activated cells from cattle and sheep in an ELISpot assay using the polyclonal antibodies, and we can detect activated cells secreting interleukin 17A. We then went on to use our transfectants to screen a whole panel of monoclonal antibodies for species cross-reactivity against ruminant interleukin 17A. One of the things that I should have said right at the start was that it was very important to us when we embarked on the project that we were not going to produce monoclonal antibodies if there was not a need. And this is why we can use these transfectants, because we've got defined gene products to then screen for cross-reactivity of existing monoclonals. And in this case, we're looking at a monoclonal antibody against human interleukin-17A, and what you can see is that that is detecting both our cattle and sheep product in transfected Chinese hamster ovary cells by intracellular staining. So again, this is a method of identifying interleukin-17A producing cells at the single cell level. We then use that monoclonal antibody to look at the expression in activated peripheral blood mononuclear cells, and looking at cattle on the left here and sheep on the right, what you can see in the top panels is interferon gamma producing cells, and in the bottom interleukin-17A producing cells. So these are in PMA ionomycin-activated cells, so under the same conditions we are detecting many more cells that are producing interferon gamma than interleukin-17A. And what you can also see is that it's not simply the CD4 population that's producing both of those cytokines, but the CD4 negatives are. And that's perhaps not a surprise to us. Certainly for interferon gamma, we've known for a long time that gamma delta T cells and CD8 T cells can produce interferon gamma. So this really helps us now to start to identify what those cells are that are producing these cytokines and how they might interact with each other. So if we go back to our table of where we started at the beginning of this grant with our capability to analyse these different CD4 positive T cell subsets in cattle and sheep, this is where we've progressed to at the end. So the first thing is, you can see highlighted in the boxes, is that we've greatly improved our ability now to investigate IL-17A. We've also cloned and expressed the transcription factors T-bet, GATA3 and ROR gamma T either in cattle or sheep, which will then allow us to further define the association of those transcription factors with the cytokines and what the signatures of those CD4 positive T cell subsets are going to be in cattle and sheep. So, in summary, hopefully what I've shown you is that in cattle and sheep, myeloid subsets can be identified on the basis of their CD14 and CD16 expression. We don't yet know what the function of those cells are, but that is work that's ongoing. We have expressed biologically active bovine and ovine interleukin-17A, and we have evaluated several techniques for detecting their expression at the single cell level. We've cloned and expressed transcription factors that are associated with CD4 positive T cell subsets in other species, and combined with our ability now to do intracellular cytokine staining, we can look at the association of those transcription factors with the cells that we are interested in for targeting for vaccine design. And hopefully, collectively, this will allow us to advance our veterinary vaccine reagents and portfolio. So finally, I'd like to thank everyone who was involved in this grant at the Roslin Institute and at the Moredun Research Institute. I mentioned, or I had at the slide earlier, I had the names of the people in the grant, but in addition to that, a particular thank you to Tom McNeely and David Frew from Moredun, who have contributed to the data that we've presented, Kirsty Jensen at Roslin, our collaborators and supporters at Bio-Rad ABD Serotech, who've helped us at various stages of this project. Not on this slide, but I'd also like to thank Sadhana Sharma and Alex Morrow from BBSRC and DEFRA respectively for the information on STAR-IDAZ, and Madeleine Clark, who's provided me with information from the Veterinary Vaccinology Network. Thank you.
Thank you for your very interesting presentation, Gary. If you have questions for Gary, please enter them now using the Q&A button. And while the questions are coming, I'd like to take this opportunity to highlight the resources that we have to offer support for your day-to-day experiments. So, as we heard today, measuring the immune response is very important for developing new vaccines. ABD Serotech manufactures antibodies that will enable you to detect the major cell populations in ruminants in flow cytometry experiments. Among them T, B, NK and dendritic cells. And now, after today's talk, you can also better define ruminant myeloid cells. On this slide, it shows a few of the antibodies available from ABD Serotech to define these populations in cattle, and you can find these and more at abdserotech.com/vet. So one of the key ways to measure immune response is to perform ELISA on samples from animals or cell culture supernatants, and you can measure bovine interferon gamma with our easy-to-use and sensitive ELISA kit, or alternatively use our matched antibody pairs for detecting bovine interferon gamma, IL-4 and IL-10. You can also find more about these on our website. And finally, have a look at our application resource page, where you'll find comprehensive guides and protocols, hands-on tips and tricks to improve and optimize your flow cytometry and ELISA, immunofluorescence and immunohistochemistry experiments. We provide support for the whole experimental workflow from design via reagent selection to the optimization of results. All you need for achieving publication-grade experimental data. And you can find these resources at abdserotech.com/applications. And now we'll move to the question and answer session. Gary, are you ready to answer some questions coming in from our listeners?
I hope so, Amanda. Can you hear me okay?
Yes, absolutely.
Thank you.
So our first question is that you mentioned IL-1 beta and IL-12 production by the CD14 and CD16 positive monocytes. Is there any evidence that they're preferentially pro-inflammatory?
So I mentioned on the slides there that upon stimulation that they produce IL-12 and IL-1 beta. They have done some other experiments, and I'm actually accompanied here by Liz Glass from Roslin, who was the other lead on the project, where Liz and Jane Hope actually looked, and Yolanda looked at expression of these cells in animals that had mycobacterial infection. So if it's okay, what I'll do is I'll hand across to Liz to answer that.
So this is some of our unpublished research in which we've been looking at the presence of these different populations in animals infected with Mycobacterium avium paratuberculosis. And what we have found is that the non-classical CD14 negative CD16 high monocytes are present at a significantly higher proportion in peripheral blood compared to animals that are suffering from other non-infectious inflammatory conditions.
Thanks, Liz.
So in other words, they may be preferentially pro-inflammatory, but we need to do further experiments. Thank you.
Great, thanks. Our next question is, have you looked at subsets of cells isolated from mucosal tissue?
So that's quite a simple one for us to answer, Amanda, in the sense that no, we haven't in this project. So we focused predominantly, in fact almost exclusively, on peripheral blood. It was the natural, it was the easy source of cells for us to look at, but we were particularly interested in that monocyte population and in peripheral blood, so that's why we took that approach.
Okay, thanks. The next question, do you know if the expression of transcription factors such as raw gamma T by IL-17A expressing cells is the same in cattle and sheep as mice and humans?
So we don't know that. The cloning of the transcription factors was a little bit problematic for us during the project, so I don't know why, but we did have some issues with those. But as you saw from the table, we did manage to clone and express a signature transcription factor for each of those major subsets in either cattle or sheep. And at the moment, we haven't got as far as analysing those either on a molecular basis, and certainly we've been trying to work up the transfectants for screening cross-reactive antibodies. So we haven't done those experiments yet where we've looked at cytokine expression and the transcription factors. But hopefully, again, as I said at the start, this is one of these things that we're aiming to do, is improve the capability and hopefully allow us to do that.
Great, thank you. The next question. Do you think we are a little too fixated on finding the same cell subsets that we find in other mammals or models when they may not exist or overlap with other cell types?
Okay, so that is, yeah, I mean, this is quite an interesting question, and I think I probably led into that right at the start, which is this thing about making assumptions and that these may or may not exist. You know, where I think it's important to understand if they do or do not exist is if we want to take targeted approaches to vaccines, and particularly vaccine delivery systems, where we're trying to drive immune responses in a particular direction, if we've got a good correlate of protection. What I think we need to be doing is combining our, certainly the cytokine assays that we've been doing at the moment, where we've been working up the ability to look at single cytokine expression at the single cell level, is we theoretically do have the capability now, if we've got directly conjugated antibodies, to look for co-expression of those molecules, and that will certainly help us answer that question. I think that Liz might have something to add to that.
Okay, so what we do get an impression is that in cattle and perhaps sheep and other outbred species is that you don't see the very clear Th1, Th2 patterns, and that in some instances T cells actually express more than one subset type cytokine. So yeah, we're trying not to get too hung up about the subsets in other species and just simply assuming that they're going to be the same in cattle and sheep or other livestock species.
Great, thank you. We have another question. Any idea about myeloid-derived suppressor cell phenotype in ruminants?
I mean, that's a very important question. We've, as I indicated towards the end of the data that I showed on the myeloid subsets in peripheral blood, we've got a little bit of an idea of potential function with some sorted cells because of their cytokine profiles and some associated information that Liz described from in vivo analysis from infected animals. But ascribing the function is quite difficult, and at the moment I would say we're still very early in our stages of being able to look at these subsets, and I think combined with those cell surface molecules, a bit like the T cells, if we can start to look at the cytokines that they would preferentially induce upon activation, and of course a lot of that might depend on the activation stimulus that you choose to use in vitro. That might help you to ascribe some part of that function. I think perhaps looking at those in vivo or ex vivo, perhaps is a better way of phrasing it, situations where we could look at tissues, particularly in disease states, that again might help us to get an idea of some function. Liz might want to add something to that as well.
So all I was going to say is that the data that we've shown today is a start in that we've characterized myeloid and T cell subsets in peripheral blood, but of course peripheral blood is just a conduit between the bone marrow and the tissues, and it will be very important to take our work further and start looking in actual tissues and in vivo. But the issue is made more complicated because of the lack of reagents, suitable reagents. So we have to be very clever.
Okay, thanks, Liz. That leads into the next question. Where could we find the antibodies for sheep IL-4, interferon gamma, and IL-17?
Okay, so we screened a number of and we've used quite a range of antibodies in this project. So the data that I presented is actually in one of the myeloid work has been submitted for publication, and we are currently writing up the IL-17 paper. I don't have the list of all of the antibodies in front of me, but we screened a large number of antibodies that had monoclonals that had been made to human IL-17A, and we selected them from those. So as I said, I don't have it in front of me, but those will be in that publication, which is imminent.
Thanks, Gary. Another question has come in. Is there any evidence to support a phenotype of toleranogenic dendritic cells?
Well, I mean, I think certainly we've not looked at that in ruminants, so I couldn't answer that. Based on the data that we've got so far, I couldn't answer that question, but it's a good one, yeah. Okay,
just to finish off, we've got two more questions. Have you looked for co-expression of multiple T-cell cytokines at the single cell level in cattle or sheep to determine if the subsets exist?
So I think that probably relates to the question that we had earlier, in that we've done some preliminary staining on IL-17A and interferon gamma co-expression, and it appears that we do find cells that do express those, but they're quite preliminary experiments. We haven't run the gamut of the other signature cytokines, and as I said, I think that was a very important question earlier that we got, which was asking if these subsets actually do exist in cattle and sheep.
Okay, and we'll fit in one last question. Can you derive the preferential development of T-cell subsets in cattle and sheep?
Okay, so I guess this relates to the point we've just been discussing about how we might identify those subsets, but I guess more relevant for vaccine delivery systems and how you might push those. So again, we've done some very preliminary experiments where we've been looking at some of the innate driving cytokines for T-cell subsets, and we did publish data quite a long time ago, maybe about 10 years ago, where we only analysed, now this is only looking at ELISA supernatants, so it wasn't at the single cell level, but we could certainly drive interferon gamma production using interleukin 12. And of all of the experiments that we've done so far, that Th1 type driver is the easiest one that we've found. We have looked at some other cocktails for trying to drive Th2 and Th17. We haven't been as successful with those, but certainly Robin Flynn's group in Nottingham have published papers where they've looked at the Th17 or the IL-17 type drivers in cattle, and that's principally IL-1 beta, IL-6 and TGF beta, and they've done that with some success. So it's about getting that kind of cocktail right, and of course that is very important when we start to think about our vaccine delivery systems and adjuvants. Liz might want to add something to that.
So one possibility is that different myeloid subsets, both macrophages and dendritic cells, the way in which they interact with the pathogens actually determines their cytokine profiles, and that they might then drive the different subsets of T cells. And so we mustn't forget that it's really important to think about how you target innate immunity when you're designing vaccines.
Great. Thank you very much, Liz. That's the end of our questions. So it just remains for me to thank you, Gary, very much for the presentation. And thank you to Gary and Liz for answering the questions from the audience. And thank you to the audience for attending the webinar. We'll be making a recording of the webinar available.